Determining the number of cells in culture is important in the standardization of culture conditions and when performing experiments where precise cell numbers are needed. Here, we will show you how to count cells using a hemacytometer and calculate the concentration of cells present in suspension. We will also show you how to determine the number of viable cells present in your culture with the Trypan Blue viability assay.
Hi, I'm Ricky, here at Dr. Phelan's laboratory at Molecular Pathology Laboratories Network in East Tennessee. Today, I'm going to show you how to count cells in suspension and how to determine their viability.
In order to prepare the hemacytometer for cell counting, you first want to clean its surface and the accompanying cover slip with 70% ethanol. The cover slip and slide should be clean, dry, and free from lint, fingerprints, and watermarks. Next, wet the edges of the hemacytometer slightly with tap water and press the cover slip down over top of it. The cover slip should rest evenly over the silver counting area. Now we are ready for the cell suspension. To prepare the cell suspension for counting, first, if the cells are grown in monolayer cultures, detach the cells from the surface of the plate using trypsin, as seen in the video for passaging cells. Once the cells have detached from the plate, dilute them as needed to obtain a uniform suspension. Be sure to disperse any clumps in the suspension. When using the